to the context of entomology. Many people have shared their scores on Twitter, on the Twitter thread, and it's certainly a whole other game of fun to guess what the lead-up guesses are. Many people guessed it on their first or second try. Congrats to you. And I encourage you to try your luck again this next Wednesday when I release the second one on my Twitter account at IsaBetaBug. So the question, and we'll see how you fare from week to week. Okay, so the answer to this week's Wordle and its application to entomology is rather straightforward this time around. The answer is a morphological feature used by nearly all insects. Some of those who don't have it latch on to others that do. That case is called foracy. And then the ones that do have this feature only have it as adults. Some even clip theirs off at one point in their life when they're done with them. The answer to this week's Wordle helps insects move, disperse, find resources, migrate, attract mates, hide from predators, and a lot more. We are talking about wings. We are fortunate today to be joined by PhD student Robert Conroe, or Bob Conroe. I'll bring him on now. He is based at Drexel University and is working at the Academy of Natural Sciences Entomology Department, where he's studying wings and specifically the iridescent patterns found on crane flies. So welcome, Bob, to the broadcast. We've had you on in the past. It's good to have you back. I can't hear you, so we might have a technical issue. Welcome, everyone, to the broadcast. Robert Van Buren, Frank, welcome. Molly, hello. Thanks for the heart bomb. I still can't hear you. Do you have headphones or something maybe you can try plugging in? Okay, cool. Hi, Pablo. How's it going? Yeah, so today, in honor of this week's Ento Wordle, we are talking about wings. And in a moment, Bob, once we're able to get him on board with audio, he has a really beautiful presentation that we're going to screen share, which has lots of great examples of these wing interference patterns that he studies that are iridescent, something that you won't really see when you first look at these flies because they have clear wings, the crane flies. But then when the light hits the wing in just the right way, you get that flash of color. And so it's a mystery. What is going on here? And what Bob has been doing is taking lots of photographs, looking at all these different species. He can probably tell us how many different species of crane flies he's looked at so far with his research. And then seeing the diversity of those interference patterns and the different colors that show up in them. And if you're trying to put we'll see an image soon when we start the presentation. But to give you an idea, it looks a lot like how iridescent looks on bubbles or oil slicks. It's a similar sort of method that the color shows up. How's it going over there, Bob, with getting... Oh, no. Maybe if we really have to, you can call me and then talk that way. And then I can just have you talk through the phone. Can I unmute on my end, possibly? I don't see a way. Hello, we're on Twitch. Hi, Dan. How's it going?
How about now?
Oh, yes. We have sound. Woo! Excellent. All right. Okay, so I will bring up the presentation so we can... Not but like when I went to bring it up, it came up on both of my screens. I wonder if I can disable that.
I hear you. Can you hear me?
You hear me?
Yeah. All right.
Oh,
Did you
great.
get it?
Okay. Oh, I see. Okay, you couldn't hear me, and now
Yeah,
it's all sorted out.
I switched a setting, and you could hear me, but I couldn't hear you, so I had to switch it back again. It was a issue between the speaker and the microphone.
Okay.
Anyway,
Cool.
Hi everybody. Sorry for the technical issues. I'm new to this service, and I'm learning.
Yeah. Wait. So quick question too. How does the sound work for our audience here? If you guys can let us know how it's sounding on your end, please let us know. It sounds you sound a little bit quiet to me, and there's a little bit of like a popping sound as well, but I am able to hear you. So maybe, depending on what people say, what they let us know about the sound on the other side, I can turn my mic down in order to get at the same audio level as you, or that way, like, there's not a big difference between the audio sound at least.
Is it any better this way? Like, is it still sounding a little bold?
It's still the same. Yeah. Here, I'll turn my sound down at least. Try that. Yeah, Bob's sound is breaking up a bit. Do you want to try calling through phone?
Sure. Yeah, let me do
Let's
that.
try that.
Yep.
Okay.
Mm-hmm.
Let's see if that works. I'll wait for you to call me, but my phone's right here and ready. Steve, hello. Sit back, relax. And I think you've seen this presentation before, Steve. But here's the new iteration. And also, this is also a celebration too, and timely, not just because of the Entowordle, but also because Bob just recently published a paper on this, which I'll drop a link in the chat for anybody interested to dive further into that. But this, you'll get the get it right from the author here on the Bug Scope. Good to see you too, Steve. Yes. Okay. I need to figure out if I can share. One sec. I'm going to do something too. Everyone taking a quick 1 minute timeout for technical arrangements. Are you ready? Are you getting ready to call me though, Bob? Okay. How to share PowerPoint. Oh, here you go. You're calling. Okay. Oh, the one problem, do you hear that? That's one issue, but we can see what that sounds like. Did you turn off your sound on your side too? Okay, cool.
Yeah, that was it was up. So there might be a slight echo.
Let me make this louder.
How's anybody? How's the sounding now?
Yeah, I'm not sure if this is better or not because now there's the whole like cellular waves picking up. Let us know, guys. It's bad. Okay. Huh.
Let's move. How about I move to another spot and see if that helps? I might be hitting like a bit of a dead spot, and maybe that's nothing.
Sure. Okay.
Bear with me, please.
I will. I'm gonna hang up soon.
Yeah, please.
Okay. Yeah, there must be an extra delay. Yeah, his mic is not working for some reason, but hopefully we can get that up and running in a moment. And yeah, and I'm hoping that I might be able to set. I'm not sure. I don't know. We'll see. Thanks for bearing with us here. Thanks for the information, Molly, about that too. So hopefully we'll get that sorted so we can get going. The other thing that I didn't get sorted out is how to make it so that when I share his presentation in PowerPoint, it doesn't take up both of my computer screens. Let me see. Click slide. Sorry. Thanks everyone for bearing with us. I'm not sure. Does anybody else know? I don't know. Oh, here. Slide settings, slideshow, setup slideshow. Okay, that did not work. Unless it's under here. I'm not sure. I'll show you guys at least what we're looking at so you can see the first slide in the meantime. Oh, yeah, I have to change that, Steve, over on Twitch. I can't hear you now. No. That's right, we're bringing you not just insect bugs, but some technical bugs today on the Bug Scope. I am using Google Chrome. Are you using Google Chrome, Bob? Okay, you are. Pablo is suggesting you may want to do a test. Oh.
Can you hear me?
Yeah. Oh, wait. Yes. And there's no way for you to talk directly into the computer?
It's not. I don't know why. It gives me the option to select my computer speakers, but it doesn't when it happens. You know what? I have a mic. Let me grab a mic real quick if you don't mind vamping
Okay.
one more thing.
Yeah, go get the mic.
I'll be right back.
This is better than before, but go grab the mic just in case. Yeah. Okay. Yeah, if we can't solve this, then I'll turn my mic down so that we're at the same audio level. That's the plan.
Let me try this first, maybe this maybe. Okay, I'll be right back. I'm going to try and switch this out.
All right. Hopefully this will do the trick. Trying to see if I can change the slideshow thing in the meantime, but I don't think I'll be able to. Oh well. I'll just watch our broadcast while I'm doing the slideshow from my phone, I guess, for now. Okay. So yeah, this is, if you look on the right here, you can see the wing interference. And that is what we're talking about. What's the deal with that, basically? So hopefully once he gets his mic, we'll get started. Have any of you guys seen this sort of thing before on insect wings? Now that it's on your radar and you have an eye for it, I bet you might notice it next time there's a crane fly or some other sort of insect that has clear wings that look pretty plain to start with. You might see something more next time you take a look. All right, fingers crossed. The setup is good with the shared screen larger on top. Hey Bob, how's it going? Do you have to select that mic on your computer as where the sound is coming from, or on HAPS at the bottom of the screen? Not sure why it's not working. All the options have worked with the other streaming video. Well, why don't you go back to what was working and I'll turn my sound down. Yeah, let's do that. Hopefully this is quieter but not too quiet. I can't hear you right now. Maybe we'll have to turn this into an interpretive dance presentation. Dance your PhD. I
Can
heard
you hear me?
something. Yes, we can hear you.
Okay, we got it. All right. So I think I got it. I have you running through my computer on the speakers, through the microphone
Okay.
on the headphones. I think we're here now.
Okay.
I'm in my son's room,
Yeah.
camping
Sounds
out, but
good.
we got it.
All right.
Sorry,
Cool.
everybody, about the technical difficulties. I'm not sure why that was doing that, and if it's still rough, I apologize for that too.
It
Issa and
does
I were
sound
literally on a
clear.
meeting like 2 hours ago, and this all worked fine right from my laptop, no problem. So I don't know why it's an issue here, but I'm here anyway. Thanks for being patient.
Yay. And there's no static right now that I hear with your
Great.
audio, so
Great.
it's even improved.
Good. I'm glad we figured it out, even if it took a little time. Sorry, everybody.
Totally. We love all the bugs. That's right. Okay.
All right. So I
I do-
guess you kind of introed me a little bit while I was futting around trying to.
Yeah. I do hear, so one last thing, I do hear a little bit of an echo
Okay.
of my own voice.
Should I turn my volume down a little?
Is the sound for you coming through the computer and not the headphones?
Yeah.
Is that what it is?
Is that any better?
Testing. There's still the echo.
Not sure. I think
That's
that might just be. What if I talk away from the computer? Does that help? Do you have echo cancel on your side?
I do, but I think it's on your side that the echo is happening. Like when I talk, your mic is hearing me. Do you have echo cancellation off?
No, I have it on.
Oh, okay. I mean, I do have it on. Sorry, yeah, I have it on too. I don't know what I just said.
Let me try one last thing.
Okay. And in the end,
Can you
it might
still
be
hear
okay
me?
because I'll mostly be quiet while you're presenting. So we can.
Oh, I got you. I hear you in my headphones now.
Okay,
Is the
and
echo
I still
better?
hear you fine. Yeah, the echo is gone.
Oh,
We're good.
all right, we got it.
All right, we have liftoff for reals now. Excellent.
Yay.
Okay.
All right. Hi, everybody.
Hello.
So I'm Bob Conroe. I guess Issa already kind of introduced me, but I'm glad I'm glad to be here and talk to you about my crane flies. So
Yeah,
I,
great
I
to.
wanted to add a little bit in the beginning, and I forgot to talk about crane flies in general. I am assuming everybody here is well versed because they're bug nerds like us, right? So they're here for the
It's a
bugs. They
mix.
probably
It's a great
they
mix.
know what crane flies are, but I'll talk about them a little. I forgot to add a lot of the background info. I should have. So sorry about that. If you don't know what they are, I'll do my best to do the elevator pitch about crane flies.
Oh, why don't you say it? Do you want to say it right now? What they are?
Yeah, sure. Yeah. So
Yeah.
crane flies are true flies, right? So they're like all the other flies you can think of. They only have the one pair of wings. They're really long, slender legs, really long antenna. Um, they are sometimes aquatic. It actually speaks to my larger thesis for my PhD. I'm trying to look at their phylogenetics, their evolutionary path through history, and understand what drove them to go from terrestrial to aquatic to semi-aquatic and back again. Possibly, we don't know. And so, anyway, I hope to find that out. But they're generally aquatic or associated with water in some way, as that when they're larvae, and then they pupate and become these really long, spindly, spidery looking things flying through the air. Um, you may have heard them referred to as daddy longlegs. In the UK, they refer to them as daddy longlegs. Here, we refer to harvestmen as daddy longlegs, right? The harvest people that the little arachnids, um,
Yeah, little pilionids,
or
arachnids.
yeah,
Yeah.
pilionids, yeah. Um, or sometimes they'll call them leather jackets because the larvae look very leathery, and they're very big. They're big,
The crane
honking
fly larva.
crane fly larvae. Yes, sorry.
Yeah.
and and here we call them mosquito hawks, colloquially, but that's not really fair. It's a really inaccurate description because while they are superficially resemble mosquitoes, they don't feed on blood. They don't have mouth parts, even some of them. So they're not mosquitoes. They don't eat mosquitoes, but we've got a lot of misnomers. You'll see them at lights a lot. They're usually pretty big, especially the group I look at, Tepulidae, the family Tepulidae, and and they can be a predominant food source in riparian environments, and not only for the aquatic animals like fish, but also for birds. So the larvae are feeding fish and other invertebrates in the water, and then as they emerge as adults and go around to make more crane flies, birds are eating them up, and they can be the sole or one of the only food sources for certain species of birds. So we're coming to find out they're really, really important for ecosystems to have there as a food source. And so they're really cool. But they serve as a food source. They break down wood. They help break down materials in aquatic environments. So they're pretty important. They just don't do anything fun, and most people don't think they're pretty. But I want to change your mind on that, at least with their wing interference patterns, maybe not their bodies, but you might not feel that way. I do. So should we get into it? I can't hear you.
Sorry, I had muted myself while you were talking.
Oh,
Okay,
okay.
sorry. I was just saying that for some reason when I click presentation mode, it's not changing on HAPS. So um, are are there any animations that will get messed up from doing it this way? Is
Uh,
that okay if we do it this way?
there might be a couple, but that's okay. I can
Okay.
explain.
Okay, we'll carry on.
Sure. Okay. So we talked about crane flies a little bit, and there's a great picture of it from our own former colleagues, well, still colleague, but not at our institution anymore, Steve Mason. beautiful picture of a pair of mating crane flies there on the surface of water, I believe, and their
Yeah.
wing interference patterns are in full display.
I'm just going to
Did you
make
have?
one other
Mhm.
adjustment so that the whole screen is in there because I see a little bit
Cool.
of it is cut off. And I don't know if that's going to make a difference down the line.
It
So,
might, probably
okay.
not, but
This will solve it. Sharing the whole screen. I have more
okay,
control.
cool.
Cool.
So,
Great.
okay, so yeah, you can see those wing interference patterns right there, like pretty blue, blue, green and red kind of colors on those wings, and the sun's hitting at just the right angle. So that'll be important to understand in a minute when we talk about what they are and why they work. So they're stable structural color patterns in the wings of insects, and they're caused by a physical property called thin film interference, and we'll discuss that. But it's a stable structural color pattern. So the structure of the wing itself gives rise to that color. It's readily visible. You don't need to use UV. They don't use scales to do it. It's just inherent on that chitin layers of the wing. And you can see it with your naked eye. You don't have to do it in a lab. You don't have to even euthanize them or anything. Like, you can just see it in nature, like in this picture. And it's the same kind of thing that you see in soap bubble iridescence, or like an oil slick on the road when there's rain. Uh, there's a main difference, though, in that those are iridescence, and iridescence by definition is shifting and changing as that surface shifts and changes. So if you imagine a soap bubble and how it kind of swirls around, the soap on that surface of the bubble kind of swirls around and it's ever changing. In wing interference patterns, they're stable, they're steady, they're not moving. So that's the main difference, but they look very similar. Okay, good, Issa. Cool. So yeah, this one had an animation. The image below it was the wing clear without any of the pattern on a white background, and so I transitioned it in to show you the same wing clear and with that pattern. But there you go. Under normal conditions on a white background, that wing would have no color on it. It would be see-through clear. You could read words on a page behind it. And that's how you expect, right? Most insects have wings pretty clear, maybe a couple of spots or some staining, but generally you can see through wings, unless they're patterned or scaled, like in butterflies and moths. Okay, good, Isekai. All right, so what is thin film interference? How does it work? So if you look on the left side here, it says upper layer, lower layer of the wing. So wings are two super thin layers that get compressed together, and then there's veins and inside of there. But that's to keep them light and to keep the wing thin so that they can fly optimally. But there are these two layers, and they're made of chitin. And when light hits that layer, most of it passes through, almost 80%. About 20% gets stuck between those layers and starts bouncing around. And then eventually it exits back out of the top or sometimes bottom layer. But when you have a black background, it absorbs all that light that would otherwise get reflected back up from the surface behind it. And then you're only seeing the light that's shooting up from the upper surface of the wing. And then the structure and the thickness of that wing, along with hairs, corrugation, folds, all that plays into how that color will turn out as it breaks that upper surface again. And it can be mapped with something called the Newton series, which I think is on the next slide. So go ahead here. Oh, no, I didn't put it in this one, but that's okay. Newton series is a series of patterned colors, and it can be mapped directly to the colors on this wing. So it's a series of ultraviolet uh, colors. It's almost a full spectrum, but it's not. There's no true red, and that'll be important as we go forward. Okay, so that's that. And this one was another a short little GIF. I slid a black sheet behind that pinned specimen there. That's Nophemia tristissima, and we'll see that in just a minute. It's a very small insect, but if the video worked, it would slide over and those wings would turn like a bluey green, and then as I slide it back, it disappears as soon as the light bounces off that white surface. So why are they important, you might ask? Well, they're a novel character, so when we ID things, we like to look for morphological characters that are distinct from everything else, right? So we want these unique things that we can tell things apart with. As taxonomists, that's what this does. It gives us a new character. So maybe we have really cryptic species that look identical, and that is one way that we could maybe parse them out because it's species specific. So this is a species level character. they're also sexually dimorphic in a lot of groups, so it might be able to let us know which ones are males or females without dissecting the abdomens out to get to the reproductive organs. And it's also sexually selected for. So we have artificial selection here, sexually, where females in Drosophila in fruit flies are selecting for a certain color type. And in that study, it selected for medium hue. So they wanted the hue to be kind of normalized, and saturation was low in that. So they really kind of were looking for bold, solid colors, is what the females wanted. Bold blue, I think, was the color they wanted most often in male wing, was a blue
That's
color.
what they were
So
attracted to.
yeah, and so it
Cool.
actually drove the lab population that they did this in, the males. It drove the population colors towards blue wings. So the
Oh,
over
wow.
time, the lab population gained overall a bluer wing in males because of this selection forced by the females. So it's definitely acting, in an evolutionary sense. We just don't really understand how yet. It's a very new thing. It has been noted for hundreds of years in literature. People noted as they describe a new species that the wing looks iridescent or rainbow or shimmery, but it's not a color. But nobody's ever really looked into it. And in 2010, Shetsova et al., I keep referencing that in this PowerPoint, they found that this is actually significant as a morphological character. So that was really cool to find out.
Yeah.
Go ahead,
So it's
Lisa.
one of those things that's been like noted and in plain sight, but not really looked into until recent
Correct.
times.
Yeah,
Cool.
yeah, it's wild. My co-author, who's my advisor as well for my PhD, John Gelhaus, Dr. John Gelhaus, he found a paper, and we cited in our paper. It was over 200 years ago, a German entomology paper that was describing a new species, and they noted the wing interference pattern.
Mm, nice. Yeah. Welcome to Blue over on Twitch is saying unknown unknown. Yep. There's
Yes,
lots of
yep, it's
unknown unknowns. In this case, it's a known unknown, which
yeah.
yeah, but
They're the
not
things
for
we
long because
know, and the things we know
yeah,
we don't know, and the things we
and
don't
not
know
for
we don't
long
know.
because Bob is on the case, figuring
I'm
it out.
trying.
You're doing it, chipping away.
So my goals here with this work, our goals with this work, were to confirm that all four of the families in the superfamily of craneflies Tipuloidea, all four families displayed wing interference patterns. We wanted to determine if there was a genus level stability of wing interference. So we know species have patterns because it's in the literature, and we saw that with ours. But we didn't know if that held up in a higher level taxonomy. And then to confirm that they're sexually dimorphic wing interference patterns in Tupulids. On the right there, I was playing around.
Yeah.
So this all started in late 2017 when I started my PhD, and it's taken this whole time to get it out. COVID and illness kind
This
of set me
paper.
back. Yeah, but this paper, I first the first pieces of it were worked on in late 2017. So that's a picture from about that time. I was trying to play around and find a good way to image, and it was quite the journey to get these imaged correctly. I used
Nice.
4 imaging systems. I reshot them all, the whole set I had like 7 times, I think, to make sure I
Wow.
got them right. It's a big learning curve, and partially because nobody's done it right. There was one study that really dug in with methods on how to capture these. So that's one place we'll talk about at the end that needs a little more work probably to standardize it. But
Yeah, lots of tinkering to find
yeah,
what worked for to bring out the features you were studying and
yep.
document them. Yeah.
Yeah, and just simple little things like glare, right? Like you don't think glare would really matter, but when you're trying to get a clean image and you don't want light obstructing it, a little bit of glare blew out most of the wing interference pattern where it hit because it was just enough light to obscure the refraction of the wing. So it
Yeah.
was really trying to diffuse the light but keep it even and keep it even across all the specimens. So it was quite the ordeal to figure that all out.
Yeah. A lot of photon managing and meddling to figure
Yes.
it out because already the subject, the interference is really sensitive.
Yeah.
So, yeah, seems like a delicate
Alina,
thing to catch.
Eliana.
Ileana.
I'm sorry, Eliana. Yeah,
Yeah.
that's totally true. There would be times where, like, I'd move it by feeling like a millimeter, and the whole thing would fall apart. And sometimes I was working with preserved specimens, so they either came out of ethanol or were dried and pinned for a long time, and so all those wings were really brittle. And when we get to how I had to prepare them, you'll see why it was a problem. So I tried to flatten them as much as possible. But even if there was like a little bend in that wing, I would get it to an angle under the microscope to take the image, and that little bit of the wing interference pattern wouldn't be showing because it's bent or folded a little bit. And so then you're like ironing out the wing under the slide, adding a little ethanol, like trying to get it to suck down and flatten. And then sometimes I did that and it just made it 100 times worse. So little
Yeah.
things you don't think about, all the tinkering we do to get our studies where they need to be
Well,
eventually.
yeah. And especially this, yeah, because every slight angle makes a difference, which also
Yeah.
makes me, not to jump to
No, no.
further questions and stuff right now, but which makes me wonder too. I think about how, like, right now, like, these are lab situations where you're photographing them, where in the wild, like, wings do have a little bit of bends and angles to them. So, what your
But
photo,
wings are
go ahead.
also innervated and full of hemolymph. So they're
Right.
much more flexible in life than they are in
Mhm.
death. So I wonder, do they have more with that muscle tone? Can they like kind of, tighten those muscles and lock the wings out and make them a little more flattened to show an image? I wonder.
Oh,
Sorry, I didn't
interesting.
mean you were
Like,
you were mid talk.
no,
I'm
no, to
sorry.
show the pattern, to show the
Yeah,
pattern.
yeah, which
Yeah.
we'll talk
Like,
about. I don't. Again, we don't want to jump to the conclusions
right.
literally yet.
Okay. Let's go to
But
next
we can keep going, and
chapter,
we'll talk at the
page.
end for sure. Yeah.
Okay.
Okay. So we looked at a lot of specimens, thousands of specimens for this, so we know that it's pretty stable on the species level. Previous works have looked at really small flies and wasps, parasitic wasps. So we're talking under 2 millimeters, under a millimeter size things. And they were clear wings, so not a lot of veins, not a lot of hairs, and no coloration. Really like glass clear, little tiny wings. And those studies got really clear imaging. And I like my images; they're beautiful. I think they're clear and they do it justice. But they seem low def, and it's because the wings are so big, so I couldn't focus down in and get a really nice clear shot. B, they're not on an even plane like a little tiny wing would be, and C, they're just harder to work with and have more folds and veins and everything. So it obscures it a bit. Um, we we looked at over 50 taxa across 4 families, several thousand, as I said, specimens, and then we picked a representative species, a male-female pair, from each family, and sometimes more than one. Um, but we looked at 25 species of Tepulidae, 13 species of Limoniids, 7 of Pediceids, and 5 of Cylindrotomids. But yeah,
Yeah. So
almost 3,000 specimens over those
and
four. Yeah,
those, I just want to
sorry, go ahead.
clarify that those are all the Topulidae. Those are all families within the crane fly group.
Yes, yeah.
And is this? These are two different wings here
Oh
that we're
yeah,
looking
yeah.
at.
Sorry, yeah. So this is a size issue. So our hypothesis, based on previous research that had suggested this, was that wings large enough would obscure the Newton sequence. That it would make that distance between those two planes too thick, and
Oh,
the
okay.
light won't be able to escape again. It kind of gets stuck in there and doesn't have enough energy to escape the membrane again. Um, and
Interesting.
and I think that's true. So we can see it here. In our results, we got 12 of 13 of the species we looked at had some wing interference pattern. The one that didn't was that top image, which is Holorusia. And I'm sorry, Issa, I don't remember the size off the top of my head. I believe it's on the wing. You can't see it either. It's small for you too, isn't it?
Oh, I have it big too. Wait, sorry. This is the top one or
Yeah,
the
the top
other
one.
one? It's 26.38 millimeters
Yeah, okay. I
in
couldn't
length.
remember. Yeah, so
Yeah.
2.3 centimeters, right? So big, big wings, and that's each wing. So you're talking almost a 5-inch wingspan on Holorusia hespera, which is the largest tepulid in North America, and it's found out west. We don't have them here, unfortunately. But if you're out west, you have them, and you probably know of them because they're huge. Some are like the size of my hand when you account for the legs.
Wow.
But in crane flies, we're dealing with this huge gap in size. So we have 2.3 centimeter long wings, and then we have 3 point something millimeter in the bottom wing, which is Nophamia, the one I showed you earlier. The whole insect isn't as big as the wing of Holorusia. It would fit within the border of that wing. the whole
Wow.
insect. So it's hard to even out this in crane flies. So in the other groups that have been studied previously, all the wings are very standardized. As we said, small, clear, no veins. Crane flies are all over the place. Small, big, all have lots of veins. Some have folds and baffling and other structures, hairs, all this stuff, and much bigger sized wings and thicker wings. So anyway, this is what we thought, and it did kind of pan out. If I was able to do more, I think I'd start selecting specimens at the upper echelon of the wing's length, wing area kind of sizing, so I could really see where does it fall out. Is there a space where a wing size precludes you from showing your wing interference pattern? So
Mm-hmm.
cool research for me or somebody else in the future. But yeah.
Cool.
All right.
Okay.
We're getting to the good stuff, everyone. So methods, we probably breezed through this, but basically that was my setup. Scope with the ring light, and the ring light kept everything even, so the light was hitting each wing the same way, parallel to the slide, and coming straight down. And then that light absorbing material, they called it flock paper from a company called Edmund Optics in New Jersey. It's used for laser purposes in like physics labs most often,
Cool.
because it absorbs 99.9% of all light, or 99.8 or something like that. I don't know. 99%, let's say. But I wanted that so I would pull all that light down that escapes out of the bottom membrane of the wing and really get the clearest image we could. And it really helped, I think. It really made that contrast nice between the black background and those bright colors. And then we did adjustments in Photoshop, but we really limited that to the initial publication that discovered wing interference patterns, set the tone. It was basically like a 10% increase in contrast, brightness, and saturation. So we really didn't play with any other metrics aside from trimming and like removing the background. And then as far as treating the actual color of the image of the wing, it was restricted to a 10% increase or decrease of those three things. So really, you're seeing basically the raw image, just maybe toned up a little bit to accentuate those colors.
Mm-hmm. Cool.
So I'll use this terminology. I'll show you where they came up. Buffington and Sandler in 2011, they looked at some WASPs, and they defined 4 pattern types in wing interference patterns. So from the top left, kind of going clockwise, we have radiform, which is concentric circles kind of emanating from the base of the wing out to the tip. And then next to that to the right is campiform, which is kind of like a solid color field, and then maybe some little spots of color coming in behind that or in front of it. Down to the right, galactiform, my favorite. They described it as I'm trying to remember the exact quote, but I can't. But as the colorful spirals of galaxies, and that's what it looks like, kind of just like pockmarked gas clouds in space, if you imagine it that way. Really colorful gas clouds in a big nebula or a system. So, and then to the bottom left, striatiform, and these are striated lines emanating from the wing origin, and then kind of following the veins out in a horizontally, so kind of across the wing this way, if you're looking at it. So just to familiarize yourself with those terms, and so far that's the only patterns anybody has found. I haven't seen any evidence of any other patterns in crane flies for sure.
Cool.
Okay, so my first goal was to confirm the interference patterns across the 4 families. And below there is our venation and cell naming convention. So in the paper we wrote on this, we described each wing in detail. And so we really had to reference what cell we were talking about. In previous papers, again, not a lot of veins, not a lot of cells, so not a lot of need to describe them. But here we have upwards of 20, 30 cells per wing. It's hard to navigate that. So we had to create kind of a map, right, that everybody could follow, so everybody knew what we were talking about. But if we jump forward, Issa, I think we're finally at it here. The good stuff. Oh, one more. Okay, so I messed that up. That's supposed to come later, but we could just jump. I forgot to delete that one. So are they in the 4 families? Yes, they are. So here's Cylindrotomidae, the first family. These are bigger sized wings. These are lost you.
I had muted myself. My bad. I was just saying, can you hear me now?
Ah,
What?
gotcha.
Okay. I was just saying,
Yep.
so you're saying when you said are they in the families, you're saying are wing interference patterns in each of the four crane fly families that you looked at.
Yes, correct. Yeah,
Okay.
that's the goal. Yep, to
Just
see if they're
clarify.
there. Yep,
All
thank
right.
you.
Sure,
Yeah,
sure.
please clarify, and if I'm saying something silly, remind me.
No, thank you for all of this. Such
Sure.
it's fascinating and beautiful work that you're doing. Yeah.
Thank you. Thank you. I appreciate that. So these are striatiform. So these striations that kind of come out from the center. They almost look like if you look at like a meteorology map of like how the wind's moving across the country from the jet stream and everything. That's how it kind of reminds me. Like they're kind of all going the same direction. They kind of curl at the end. And then if you look where those yellow bars are, that's called the chord in these wings. And it's a set of crossed veins that kind of come down vertically. And there seems to divide the pattern between the wing closer to the body and the wing towards the tip. And you'll see
It's
that throughout some groups.
it's
So
called it's called a cord.
cord. Yep. It's just like a cord, like a plug, that you plug into the
CORD.
wall. Yeah.
Okay, interesting.
And it's because it's
Could
kind of,
could I was gonna say could be a future Wordle. Chords.
oh, yeah.
Five
Yeah.
letters right there. Hint.
But it's not an H, I don't think, unfortunately.
Oh,
But,
add an S and we're good, I think, right?
oh, yeah.
CORDS.
CORDS. Yeah. Yep,
Wings,
you're right.
chords. Yes.
despite that showing, I have a perfect Wordle score right now. Just absent-minded in the moment, apparently. Anyway, so yeah, this chord is really interesting, and it's because the wings are so big. So there's so much area to the wings, and they're very long. So it's kind of a structural reinforcement there. All those cross veins, and there's a little bit of thickening there in the wing, and it creates a stronger wing in that sense. So, but it's really interesting that the pattern changes before and after, and I'd love to investigate that more. A lot of it has to do probably with, I would guess, body weight and body size, because you need wings that are able to lift your body. So as you get bigger, your wings tend to get bigger. But I also think you'd want your wings to be light. So maybe there's, some give and push and pull there. And when you get cut off from showing your wing interference pattern as a crane fly. If you have thin wings, but you're big, maybe it shows, because cylindrotomids are not small. They're not Hularusia, but they're decent sized insects. They a lot, and they mimic a lot, so they tend to look like other things. So I don't know so much here, but definitely have the wing interference pattern, and it's pretty clear. So okay, we'll jump to the next family, the Limoniids. So this is that Nophamaya I showed you in the beginning, and we noted how small it was before. And here you can see too, it's very small, a couple millimeters long. And so females are always on top in these images. As we go forward, males are always on the bottom, just for reference. And you can see here they're definitely sexually dimorphic. And this is the campiform pattern, in my opinion. That's how we defined it anyway. I guess you could argue it's galactiform, but it tends to be a really solid background. The females have a little bit of variation. You can see that yellowy green band coming down in the middle there, kind of. But the male is clearly campiform, a solid green field with little bits of red and blue. Franklin, no, it's not in ethanol when it's taken. If we go...
Yeah. I was going to say the question that Frank is posing is the specimen in ethanol while taking the picture, for anybody who's not able to see his comment?
Oh yeah, no, it's actually just pressed down between a slide cover and a slide. I use ethanol when I seed it in there, like when I set it, so that it sucks down and hopefully sticks to the bottom of the slide. And then I press all the air out with the cover and glue the cover down around the edges. And in doing that, hopefully get all the air bubbles out and flatten them and stretch the wing as much as possible.
Wait, so there is ethanol in there, or it dries out? Or
no,
what?
So it's really high concentration ethanol, so like 90%, okay. And it's just in there to kind of, it wets it for a moment, and then it flattens, and hopefully I get it flat and even, so that when I put it under the scope, the light hits that plane of the wing evenly, and we don't have those issues of dropout, as I mentioned earlier, where there's a corner of the wing that's just clear and the rest of it has pattern. So no, but they did, one thing I forgot to mention, these did come, I think, from both pinned and ethanol. So some of them were stored in ethanol for years and years and years, and then they were removed from the ethanol. I excised that wing off. Under the scope, I remove that wing, and then I do that process to set it down on the slide.
Um, while we're on that topic, like, do you know what some of the oldest specimens that you looked at? Like, do you know how old some of them
Early
were?
1900s. I want to say 1904 might have been my earliest one. I
Wow.
didn't want to go much earlier because I didn't, it just, even the 100-year-old ones were in rough shape, right? There's not a lot of
Mm.
wings that don't have a nick or a chip or a tear in them after 100 years. So unfortunately, they're perfectly fine specimens for the museum, right? They're still intact, but I needed really pristine wings, and unfortunately, age limits the number of pristine wings you have. So
Mm.
um, and a lot of times, insects damage them in life, so by the time you even catch them and preserve them, the wings are already somewhat damaged. If they lived a long life, I've seen insects already that look, they look like a tattered flag by the end. Their wings are just,
Mm-hmm.
shreds by the end, fluttering in the breeze. So I did use some old ones though when they were of a sufficient quality to see the wing interference pattern. I would have gone older if I
Cool.
could, but we couldn't facilitate that.
Sure. Yeah. And I wonder if it's, well, I guess for your purposes it would have been fine, but I guess I wonder if you can detect much variation between ones collected so long ago and today. Like if there's a
No,
significant difference.
so our study initially was to investigate that,
Oh, okay.
and it didn't work at all. Like, we did the studies, we took alcohol specimens over a span of age and pin specimens over a span of ages in the same species, and lined them all up, like imaged them all,
Mm.
looked at the wing interference patterns, and just no difference.
Okay.
It, I think if it dried out in a really rapid way where it may have deformed the shape of the wing, then you might have an issue. Like, if you really did like critical point drying on a specimen, that might change the overall dimensions of the wing in a way that would
Mm.
change how the light would pass through it. But that chitin is hard. I mean, chitin doesn't really change chemically, so I think it's pretty stable unless you break it.
And chitin's what the wings are made of.
Correct. Yeah. Yep.
Yeah.
So
The proteins. Cool.
yeah, so I don't think age or preservation has any impact on it, but we didn't fully investigate it. But because we kind of had no leads when we tested it, we kind of just left it at that. And we saw no evidence that there was a changeover. At the academy, I'm sure if you've seen Issa's scope before, she's probably shown you our drawers full of specimens, and sometimes they're really full. And if you tilt that drawer just the right way, you can get the wing interference pattern to shimmer
Mm.
in the light.
That's so cool.
And so I'd have full drawers stuffed with specimens pinned from all different ages, all different locations across the country. And when you do that, even under the scope, if you put a couple of them on a tray under the scope and do it, they all shimmer in unison if they're on the same plane, and they all look exactly the same no matter how old they are. So really, really
Okay.
stable colors. Yeah.
That's so cool.
We were worried about it early on that this was some transient thing that wouldn't be, stable to ID with, but it really does seem like it's stable and strong. It's really clear.
That's really cool. Awesome.
So, all right, let's
Next
jump to
one?
the next one here. We'll kind of. Oh, I've had some issues here, but this
Oh,
is
I see. Yeah.
Pediceidae. You might be able to
Mm-hmm.
just like shift it up over the
Shift it.
word at this point. Yeah, just a little bit, but
Or I'll
um,
make it a little smaller maybe. Oh no, unless
yeah,
there's
there
um
might be something under it. I don't know.
okay.
I think it's okay. I think the point I need to make is seeable here.
Okay.
But these are definitely galactiform. They have these big splotches of color kind of all over, and the male and female are clearly different here in a vastly obvious way. If you look at the wing tip on both of those specimens, kind of the bottom right of each wing, in the female up top, it's a pretty nice golden green color there. And then on the upper edge of that wing, there's that red band. Um, and then if you go down to the male in the same spot, that blue, dark blue purple in those same cells, and then bordered by a green yellow, and then the red is pushed up the cell higher than it was in the female. So it's this really,
Quite
really
a rainbow.
intense, different rainbow pattern on each wing. And again, you see that dark blue. I can't say with any impunity, because we didn't test it, that dark blue means something in the male wing. But I suspect if I studied this group, it might really matter, and that might be being selected for in the male, that dark purpley blue circle there, kind of on the lower right side of the wing. And there's a secondary one kind of further to the left from that, down into the left, and that's also different in the
Right here.
female. If you look, yep, thank you. If you look up at the other image, totally, different pattern, different color. So
Yeah.
that's really promising there. If I was to go back, I might go after Pedicea, especially because they're really common. Pedicea and Constance that's the species. If I wasn't clear,
Yeah.
but again, this is our galactiform, and like I said, we didn't see radioform in our groups at all, so we won't see that one. I don't know why. It might be a size issue that most of these wings are long and thin as opposed to short and round. Um, so that might just preclude radioform from developing. All right, let's jump ahead here, Issa. Oh, yep. So looking at sexually dimorphic stuff. Actually, Issa, I think we could just skip this one. I don't know
Okay.
what was going on here. I think I probably forgot to delete those circles off from another
Oh, okay.
from another time. All right, we can
Yeah.
jump again. This was just to keep us on
Okay.
track. Yeah, the generic level patterns. So there's some animations here, but I've highlighted the 3 male wings of 3 species from the genus Nephrotoma. And I was trying to look at, do males across a genus have a similar pattern, or do females, or do species in general have them? And granted, our work in this was limited, but we saw no evidence of this either. So if you look at the 3 squares, that's our 3 male specimens across 3 species, and there's no real congruency there aside from the color. But all of them have magenta and red and green in their wing interference pattern, almost all of them, or the blue greens, but that's not unique to Nephrotoma. So we didn't see any evidence of a pattern amongst all these different species in the genus. So we're thinking, no, there's not a generic pattern. But other research has kind of suggested maybe there's something going on. So it really, I think it's just going to take a big, widespread study of a single genus and get as many specimens as we can. And then doing something we'll talk about at the end, which is a human independent sampling of these wings. We can take our biases out of it and just kind of run it through a computer system, an algorithm, and just assess it in a statistical way, not in a kind of subjective way that I'm assessing these colors, because what I see and what you see might be different, right? So we have to standardize that somehow to be more scientific. But this was just to make sure it was there. So if I'm able to ever come back and study it again, that's absolutely, I think, what I'm going to go for.
Cool.
And the females also, no pattern there. So okay, so we got those 2, and then you can jump one more, and we just to confirm sexual dimorphism. We already talked about that a little bit. Definitely there, and it's there in other groups too. If you click ahead, the next one, there's the Pedicea again, and then I think there might be one more. Nope, it's not. But this was an interesting thing. So in our quest to look at that change within a species, we took multiple samples, multiple specimens from a species to look at. None of them had any variation in them. All the males looked almost identical. All the females looked almost identical. This is this Nophamia tristisima again, always causing problems for me. The 3 top ones, A, B, and C, there are female wings, and the 3 bottom ones, D, E, and F, are male wings. And the animation won't come up, but the 2 in the middle, if you look at C and D, they look almost identical, in my opinion. They look very similar, and there's no good reason for that. Males seem to have some males within them that have female leaning wing interference patterns, and some females seem to have male leaning wing interference patterns. Not really sure why that happens. My hypothesis off the top of my head is that it's having to do with size variation, and that's really all there is to it. And so if males get closer to a female size, they tend to have female style wing interference patterns. And if females get closer to male size, they tend to just have that. But there's not a lot of size variation between males and females, so that doesn't really explain the whole picture. Not really sure what's going on here. We were not able to figure this one out for the paper, so this could be another paper in the future too. But really interesting, and it's interesting that it was this one group. We really saw nothing like this in any other group. So I don't know. Big question, but hopefully we can answer it someday.
Totally.
And then I think that's just about it. Oh yeah, I did do it. Okay, there you go. But
This?
conclusions, that's okay.
Oh, yeah,
It was just
okay.
blowing up those two middle ones to show that they were looking similar. So
Okay.
this is first family level survey of tepulids. I'm proud of that. Tepulid wing interference patterns.
Woo.
We can confirm the wing interference patterns across all 4 families. 12 of 13 of the species we looked at had visible stable wing interference patterns. We can confirm sexual dimorphism. 8 of those 12 species that had a pattern were dimorphic. 4 were similar. So it's not a foregone conclusion that wing interference patterns are sexually dimorphic in crane flies, but there are sexually dimorphic wing interference patterns in crane flies. It's not a prerequisite. And they don't appear to have a stable generic level pattern. So on to what this all means for the future. The fun stuff Issa and I were talking about earlier. So moving to a viewer independent assessment model, Butterworth et al. 2020. This was a group of researchers from Australia, and they looked at sarcophagid flies, and they were able to use a computer algorithm to map out portions of the wing and break it apart into 5, I believe, sections. And then they assessed the color pixel by pixel in each of those sections using this algorithm. And then created a statistic based on that to compare is section A of the wing, similar to section B, and then you can take that whole number that they get out of all those sections when they calculate it all together and compare wing from species A to wing from species B and do a viewer independent thing and say, yes, statistically these are differently patterned, differently colored wings. And that's what I'd like to do because it takes that bias out of it. My co-author and I and other people in our lab, we not argued like yelled, but we discussed heavily about this because there's so much there, and we all had different opinions on it. We all thought different ways about it, and you get to that that point where you can't agree, and everybody's kind of like stuck into their view, and then you just kind of make a decision. It would be great to just remove that, right? I mean, we all get along, so it wasn't an issue. We resolved it peacefully. It'd be great to not have that issue. And then there's no questions of validity later on, right? You can't come back and say, well, Bob was losing his eyesight when he wrote that paper, and he didn't know it at the time. Or something crazy like that. You don't know. So, I mean, I just want to, for the sake of science, you want it to be as unimpeachable as possible. So moving forward, I think that should be the standard when doing wing interference patterns work, is to have something independent of your opinions assess it. And then if it's sexually selected for in crane flies, which there might be some evidence of that, why? Because the vast majority of crane flies, the males kind of mob the females. They actually will sit and wait outside of the area where they know they are. They can smell the pupa in the soil developing, and they'll come and hang on all the vegetation around the area where these things are. And as soon as they emerge and dry off, the males swarm them. Imagine a fight scene in a Looney Tunes cartoon, like that big cloud, and you see like arms and legs coming out. That's kind of what it looks like. You'll just have 50 males converge and chase down this newly emerged female to mate. So she's not really selecting anything in that instance. But there are some species where males do either gifts or dances or compete in some way for the female. So maybe in those instances, that's where we're seeing the dimorphic or sexually selected for patterns.
Did you happen to notice any trends there with the species you looked at for this study? Like knowing their behavior, or did you not really try that up?
No, because the one I think the one I showed you does not have. They have the lekking behavior where these males congregate and chase the female. So and that was some of the ones that had the spots that seemed. Yeah, like Nophomia and the Pedicea, they both have that trait in their reproductive strategy. So there's no reason that the female would select for anything. But we just don't know, right? And there's 16,000 plus cranefly species, or 5,000 cranefly species. So there's a lot there to pick through. It's hard to choose. So there's a lot of work to be done there. But and then a larger generic level study, as we had talked about, I think that would be important But the coolest thing I discovered, and I have no evidence to this, but I wonder, and this is where my brain goes with it, is could wing interference patterns be camouflage when resting on the leaves, as in the pictures to the
Hmm.
right? So especially if you look at the bottom left image, that's Nophomaya again. They're mating, but you can see
Wow.
with the wing interference pattern active, they kind of blend into that leaf pretty well. And
They
if you were an
do.
insect with less than optimal vision, I don't know that you'd notice that that was an insect there or a pair of insects. So I wonder if they're doing it as a sort of camouflage or if that's the default state of wing interference patterns, just to break your pattern up when you're hiding or resting somewhere. Uh, or could it be dazzle, flash, or startle camouflage? So this is a newer function that people have discovered in some insect groups where they have something metallic or iridescent or shiny, uh, and they have a mechanism either on their legs or their wings or some other appendage that they can quickly flash it or otherwise make it change, and that flash, that spark of light, of color, of shimmer is enough to distract your predator to allow you that extra millisecond to get away from their jaws. And being perennial prey items, I wonder if crane flies were, if this was selected for in crane flies as a defense mechanism, that those that had sufficiently thin wings were able to display a wing interference pattern which camouflaged them and made a differential reproduction where they were more successful than their counterparts. And over evolutionary time, did that grow into these wing interference patterns? The only caveat is when you have purple and yellow and blue and red in there, you know, if you're hiding in the dead of summer, there's probably not a lot of those colors about on leaves. So, but maybe again, like I said, it's just to break
Flowers.
up the pattern. Flowers, I didn't even
Yeah.
think of that. Crap.
Pollen. Yeah.
Oh, well,
Petals.
that's why you're here, Issa, to keep us honest.
Brainstorming. Anyway,
Yeah. So I think that's about it. If you
cool.
tap forward, but I think it's just my notes, my thank yous to
Yeah.
everybody. So thanks to everybody
Yay.
who helped me. Issa, I don't know that you reviewed my paper, but if you did and
No,
I
I don't
didn't
think
put
I
you on
did.
here, I'm
That's
sorry.
okay. It's okay. No, no.
But thanks to all these groups that helped me, and then I'll definitely take questions from the audience or from Issa or from anybody.
Yeah. Yeah. Thank you so much for
Sure.
sharing this because it's really cool work that you're
Thank you.
doing and dazzling images and everything. Um, and I just want to go back one because that image, see, is just such a cool image. I feel like there must be some camo involved. And um, yeah, it's cool to hear about that different work happening. It also just makes me think about like um, iridescence in general showing up in different wings and whatnot. Um,
Yeah.
because there is that flash and dazzle like that morpho butterflies have, for example, that you
Right.
guys
Yeah.
were talking about, where they look camouflaged and all of a sudden they open their wings and it disorients the predator. Um, and um, I've wondered, like, I'm very curious about what variation there is, like, in between the different species. Like, if you were to do the pixel variation, because I'm sure it's like a face, like, there's little differences from one individual to from the one to the next. Um, and then, like, I wondered that, for example, with the Madagascar sunset moth, because I've noticed that, like, they are like a full, they're like kind of full iridescence in a way of all these different colors and shiny scales of different colors. And they have a general pattern, but it just seems like pretty variable from one to the next.
Yeah.
And yeah, I wonder
I
about that.
I think because I looked into this, so the scales on most lepidopterans are uh, multi-film interference. So they're like, we're thin film interference with wing interference patterns because it's just the two planes, but with the scales they create like lattice work of planes, and then the light's constantly bouncing across like a diamond, you know, so it's more
Oh, okay.
shimmery than wing interference pattern
Interesting.
because it's extra layers and angles and stuff. So
Cool.
yeah, so I do wonder if that
Yeah.
I saw some questions. Do you mind if I
Yeah, go ahead.
Answer?
Just
Okay.
read them out loud
Yeah.
before you
So,
answer them. Yeah.
Ileana, what's the number one thing you'd want everyone in the world to know about what you've learned in your work? That's such a good question. um, I I guess. Well, I'll give you two answers. I'll give you the science answer, and then I'll give you the metaphysical, philosophical answer I have for that. And I think scientifically, it's that there's always something new to learn, right? That you should be looking closely and abstractly at whatever you study. And that doesn't mean just insects either. I guess that really does extend to all of our work, but especially in science, we need to be thinking outside of the box, and you almost have to break your brain into doing that, because our brains are logical, right? We want to find patterns, we want everything to make sense, we want everything to complete, and we can't ever do that because the world isn't nice, right? It's not fair, and it doesn't work that way. It's chaotic. um, but keep looking at things in that new light, because this could break open a lot of taxonomic mysteries of things that look the same but are distinct species, or groups where we don't know the female because we haven't seen it, but it might be hiding in our collections as another species. But if we knew what the wing interference patterns look like, we could look for that pattern in females that we don't have. So, and that would be true for a lot of other work too, right? Just look at your work in a new light. Try and look for that weird thing that you know isn't going to work, but hey, screw it, I'm going to be late for dinner and look at it anyway, because that's where these great mysteries get solved. Is somebody saying, yeah, I am going to take that extra time and just putz around and look at this silly thing under the microscope.
But don't miss dinner too many times. Work-life
No,
balance. No, but yeah,
no,
totally.
no, not a, yeah, exactly. But you know what? Like, I wasn't personally interested in this work. My advisor from my undergrad sent me the paper, the initial paper that this is all based on, and it just got me excited. And I just curious. I didn't even touch it for like 3 years. It just bounced around in my brain, and then finally one day I got that urge, um, and I did it. But I will say, I guess I kind of answered the metaphysical, philosophical one too. It's like to just never stop that exploring, that wonder and curiosity, because you don't know what you're going to find. I hope that answered it.
Mm-hmm.
And then
That's great.
Frankman asked, "Do you always see the same colors with different photo equipment and light?" Um, yes, though I didn't play with different like temperatures of light, so I don't know if I used like a really warm light if that would change it, but I don't suspect it would, because light's light. The color might be a little different, but I don't know how that would work. But we use just plain old white light, so it didn't really affect it too much. And then he also had a follow up, Frank. How wide ranging is this effect species wise? In what we looked at, the vast majority of the 50 some species we used in this study to examine all had it. So it's pretty common. I think size is the biggest limiting factor, and having wings, of course, that are clear and not obstructed with color or hairs or something like that. So I think you would expect to see this in the vast majority of cranefly species, excluding very, very large ones. But that's really the minority. While craneflies are big, most of them aren't that big to preclude them from having it. So when I was early on working on this, I would just go through the collection and like pull a drawer out and just look, and and then I'd find ones that I really like the pattern. I'd pull that species out and take imaging, like a series of images of that. And I saw it pretty readily in all groups. Some of them were just boring. Some of the color wasn't that interesting. So I think there was where my bias kind of showed a little bit. I picked ones that I thought looked cool to begin with and rolled from there. So that's definitely something I'll do different in the future too. But you got to start somewhere.
Can you see it? Frank asks, can you see this wing interference pattern outside of crane flies also?
Yes, yeah. So most of the work has been done outside of crane flies. This is the first study to really focus on crane flies. The initial paper that came out in 2010 did image one male crane fly for their paper, but they were surveying all the flies and all the wasps, all the small parasitic wasps. And so there's some evidence there, but it's also in um, in scale insect, the males are winged, the females are wingless, but the males have wing interference patterns, and they're really hard to identify to species. But using the wing interference pattern, a paper that came out a few years ago was able to identify males and segregate them to species level, which is really cool for that group. Um, and then there's some other work in other fly groups and other wasp groups as well. Nobody's branched out much further than that. It's mostly been flies and wasps, and then this one lower hemimetabolist group. So I'm curious to see who else has it. I will say I know just from seeing pictures, high definition pictures, Dermaptera, the earwigs. If you didn't know, earwigs have wings. They're folded up in those two little squares on their back. Hey, buddy. And they're beautiful. They're huge, papery, leathery wings, and they fold like one of those rain ponchos that you buy at a gas station. It folds up into in your wallet. It'll fit in your wallet. That's how those wings fold up in Dermaptera too. So they have it, though nobody's published on it. I've just seen it in pictures, so I know it's there. And I know things like ants, the winged reproductive ants have it.
The alates.
Yeah, and I suspect most other wasps and bees of a certain size would have it too. I don't know about other groups. I know dragonflies don't have it. Cicadas don't have it. It's the way their wings are made. They have a layer on the surface that kind of blocks out the light from transmitting very well. They have iridescence on the surface of their wings, though, so it's a give and take.
Cool. I do have another question or comment as well about, like, it'll be cool if you, remark, I suppose, to come across while you're doing this work and continue it, like an andromorph, where you discover it's a gynandromorph because you recognize the two different wings patterns of the male and the female on either side.
Well,
that'd be
I
really
will have to come back
sweet.
on for that if I ever discover that. That would be so cool.
Yeah.
I actually, do we have any in the collection, Issa?
We do have a gynandromorph in the collection. For anyone who's like, what are you talking about? The way that insect genetics work, which I think, Bob, you understand them a little more than me, especially thinking of your Twitter handle, 1/2 Lloyd Hart.
Oh, yes, yeah. So especially, in wasps, right? The male only has half of a chromosome complement. So he's only giving one set of chromosomes to give, a half set. And the female gives her half, but she can give either half. And sometimes they don't fuse, right? The gamete, like the sex cells don't fuse, and so they develop as a male haploid and a female dip. Basically, it's almost like facultative. That's getting into the weeds. I don't need to talk about that. But it's almost like she's being parthenogenic in a way, because the female reproductive cell just starts dividing, right? And then it basically develops two haploids that are smashed together in the middle.
Yeah, so I don't fully understand it myself, but what I can say is that the genetics of insects are that there can be instances where half of the insect, like if you cut it in half and half of it, like if you were to mentally cut it in half, if they're not bilaterally symmetrical, one half is male, the other half is female. And it's especially remarkable when there's sexual dimorphism and the male and female look different. And for example, if you look up sexually dimorphic or gynandromorph cardinal, that's especially striking because it happens in birds too, where half the bird will be bright red, the other half will be like a more mellow, muted coloration. It's less obvious, of course, with animals that are not sexually dimorphic. Like with the crane flies, they're not always apparent, like first glance color wise necessarily. I mean, that's changing with Bob's research now, but opening our eyes. But they're not like off the bat sexually dimorphic in colors. You can tell because they're tapered abdomens, which actually, this is a great example up here with A and B. B is a female with a pointy abdomen, and then A is a male with claspers. But then with the work that Bob's doing, like getting familiar with wing interference, if you see it, you might be able to tell the difference right there with a whole different way.
We're lucky with crane flies because they wear their genitals on their sleeves. They have external genitalia like we do, so it's really easy to tell which one's which. Most flies don't. Most flies and most wasps, even if they're not sexually dimorphic, you have to dissect out their genitals from inside of their abdomen. It's a process, and it damages the specimen. Obviously, it's not a complete specimen then. But if you need to know, you need to dissect them. So we are lucky, but it would help a lot of other groups if they could tell which ones are males and females without dissecting them. It means more specimens in our collection that aren't dissected, that are full specimens for preservation. So it's really important. And that's one way, actually, that I think this could really benefit in a way that you wouldn't see it at first, right? Like, that's not a logical conclusion, I don't think, from crane flies have colored wings, that change based on the angle. I don't know that that's a logical conclusion to say, oh, we can tell, we can do this now. But it's something that may come out of it, that if you have a group that's hard to tell. For example, I've been working on tachinid flies, little parasitic flies, and they all look identical, and they had to dissect out hundreds and hundreds of genitalia to tell which one was which for this study that they were doing in this material I was working with. And it's tedious, it's time consuming, and it damages the specimen. So right there and then it improves our collections, our museum collections of insects, by not having to damage as many. So it's a really good outcome of all this, I think. We just need to get our community to adopt wing interference patterns now, because the vast majority of people don't know about them, or don't think they mean anything yet. They think they're just pointless still. So it's a kind of a PR battle to get wing interference patterns to be appreciated.
So hear ye, spread the good word of the wing interference patterns. And I mean, those colors, those photographs you got are just dazzling. So that's definitely gonna help with that initiative.
Yes,
Kind of
yes, for sure.
Question that goes along with what we were talking about a moment ago from Frank is,
yes,
Frank Mann, are the colors and patterns exactly mirrored between left/right wings?
Generally, yes. I don't think you could tell with the naked eye that they're different, or at least not on a quick glance. But when you go kind of cell by cell or band of color by band of color, there's absolutely variation among specimens, right? So from one male to the next male, that line between the red and the magenta and the green might be different, right? It might be shifted down. It might not be a clean line and from one specimen to the next, but from one wing to the next, it's pretty standard. There is some variation because each wing develops on its own, right? And so the chitin might be thinner on the left wing than the right, but the overall blueprint is 99% similar, right? So you're not going to see a lot of variation. It's mostly going to be little speckles. If you think about static on the TV screen, right? Like if a couple pixels went out or went different color, you would notice them, but they wouldn't. I don't think you could see that they were inherently different.
Or I have a freckle over here, but not over here.
Oh yeah, exactly,
exactly.
Like yeah. Or how our ears aren't the same shape on one side or the other, but they're ears, right? They look like ears. They don't. One's not a nose and one's an ear. That would be really confusing. But and the same with these wings, right? There's subtle differences, and if you scan them and put one next to the other and lay the image on top of one another, they would not be 100% the same for sure. I don't know how much different. That's again one thing we didn't quantify in our study, and that's why that Butterworth study, the human independent one, is really important, or bias independent one, because they take that out of it, right? That I don't have to guess if it's the same or not, or same enough that it matters. I just let the computer look at it and give me that readout. So that's why it's really important, I think, to shift to that in future work on wing interference patterns.
I have a remark to say, and then a question for you, and then we'll wrap it up because we've been here for a while now.
Sure, no problem.
The time and everything.
And thank you all for sticking with the technical issues. Apologize again for that.
I'll edit that on the YouTube version of this.
Oh,
nice. I can cut it out on the YouTube version. So
I'll look cool then. I won't look like an aging millennial who's forgetting how to use technology.
So I wanted to say that I think that I'm so curious about the idea of the wing interference playing a role in camouflage, not only because that photo that you showed but earlier, but also because I do think that iridescence does break up shape. And I have a personal example of that, and that's that. So like I have the bad habit of like tearing off my fingernails or biting my
Hmm.
fingernails and all of that.
Me too.
Cheers. um, so I usually don't really wear any nail polish or anything like that because I mean I could say it's because the environment and nail polish is not good for it. But you know, sometimes it's fun to wear some color, a splash of color. Um, but if I do put something on my fingers, I usually choose something iridescent
Hmm.
because it doesn't look, my fingernails don't look as bad because I think
It's
it breaks up the
distracting
shape
from the
more.
pattern. Yeah,
Yeah,
you're seeing the color, not the pattern, right?
yeah,
Yeah.
you can't see like the clean cut lines as well, or like, you know, all the irregularities of my fingernails not
Or how much
being
the
trim
finger
and
skin bends over the nail because you've chewed it down so far. Or is
anyway,
that
but
just
yeah,
me?
I don't know. Um, but it's something I definitely want to break because it makes it easier to point to bugs during outreach with my
Yeah.
fingers when they're not torn. But
Everybody says to use the sour stuff, but I don't. I think I would just adapt and
yeah,
learn to eat the sour spray you put on your fingers so you don't bite your nails.
oh yeah, I
But
just
it's
have
what, the
to
30-day thing, right? You got to not do
commit.
it for 30 days.
Oh, well, once I almost, not to go on too much of a tangent, but once I did almost break out of it, and I was like, oh, like, what difference does it make if I do? Or
Yep,
something
that's how I
like
always get to it.
that.
Or you get super stressed out and then you forget that you're
And then it
not
goes
doing
to
it.
ground
And
zero again.
yeah, yeah, it's
Anyway,
hard. I
but
quit
yeah, so,
smoking, so I'm holding on to that one.
oh,
I quit
that's great.
smoking now
That's
for
good.
almost 10 years, so
Oh,
doing
fantastic.
good.
Oh, Steve joining the
Exactly,
club
Steve.
here.
Exactly.
Saying it's so tempting and easy to do it when your nails grow out. Yep.
Yep, that just happened to me actually. I grew them out around the holiday, and then
Oh, nice.
beginning of January, I was like, "Wow, these nails are really long. I better do something about
Trim them.
that."
Yeah.
I trimmed it
Anyway.
with my mouth though. So anyway,
Oh,
back to
yeah.
your question.
But yeah, so I find that iridescent or shiny nail polish works best in those situations, and I don't know. And so it just makes me like really think that there's something there about breaking up the patterns and the shapes and
Well,
being a
you could
little
do a
more
study.
amorphous that way. Yeah.
Yeah, you could do a study on this. You could paint your nails with iridescent and non-iridescent paints, and or a clear coat maybe, and then
Yeah.
get insects and slowly bring your finger up and like try and get it to shimmer and see if it, we could test this out if it
Maybe.
works.
Or just ask people to rate how the attractiveness of my fingernails with opaque colors versus iridescent colors. Although then there's like the shiny bias, but people who like shiny things, but
True,
that
but
can be
maybe
controlled
insects would
for.
too.
That can be
Yeah,
controlled for in one way or another with
we're gonna be out
with
there in tinfoil collecting this year, trying to calm
yeah.
all the bugs down so they just sit there and we can grab them with our hands. No more nets, just foil.
Yep.
Anyway, you had a question too, you said.
Yeah, so coming back to our topic here, I was just asking like what other projects are you working on for your PhD and where are you in that process and what are you looking forward to in the
So I'm
years to
in my
come or
fifth
months
year
to
of my
come?
PhD. So I'm in the middle of it right now. I should be closer to being done, but of course, COVID has set us all back, and my work too. Um, and so I'm getting there, but that main project is a phylogeny of the family Tepulidae. So some issue with naming. So traditionally, all crane flies were under one family called Tepulidae, and then it was found that's not really what reality is, and so they split them into four families. But some people don't like that. So there's a family Tepulidae, and we say in the strict sense, meaning just that family, and then there's the family Tepulidae in the broad sense, which is now four families, depending on who you ask. Most I think crane fly workers think it's four, but there's some that still stick to the one or a different one. Um, that being said, sorry, Issa, could you repeat your question? I lost my train
Oh,
of
just
thought.
like what else are you working on with your
Oh,
PhD and
yeah.
looking
Sorry,
forward to
I
in the
got my
next
brain in the weeds.
year or
Yeah.
so?
So
Yeah.
so anyway, my work's on that one family, Tepulidae in the strict sense, and I'm looking at their phylogeny, so how they evolved over time and how we got the number of species we have today, and how they're all related to one another. Nobody's done that. so some people have looked at smaller a genus or maybe some species and map them out in a phylogenetic sense. But on the family level, we have no idea how these genera are related within this family.
Hmm.
Some vague relationships, but we really don't know. So my goal is one to do a molecular phylogeny of the family, where I'll sample about 150 of the 4,500 specimens in Tepulidae, and I will take that subset and use that as a model, right, to look at their evolution. And then once I have that model, that tree, I can apply different metrics to it. And I want to see when in evolution in evolutionary time did things become aquatic or move to semi-aquatic from aquatic or move to semi-aquatic from terrestrial or become terrestrial, and so we don't really know a lot about that either, but I'm curious how time and evolution affected that. Is it that they kind of shift based, sidewinding as things happen, things, maybe a species was aquatic and then it moved to semi-aquatic as things got dry in its environment, drier, and then maybe even eventually shifted to land. But we just don't know. So I'm really curious about that and to see when these times diverged. And then lastly, the last part of that research I want to look at is the larva. So we already know a lot of traits that are for aquatic larvae of crane flies, and we know traits that are maybe more terrestrial. And I want to look at those traits and compare them with this new phylogeny we have and see, okay, this is a semi-aquatic species. It has aquatic. Does that mean then if we map out in deep time, was there a transition where it used to be aquatic and it shifted to semi-aquatic and kept some of the traits of an aquatic
Hmm.
larva? We just don't know that. So a lot of that will shake out of
Yeah.
this initial phylogeny.
Will you define semi-aquatic?
Sure, the best I
What?
can.
Okay.
Um,
Curious.
so semi-aquatic means like aquatic adjacent, or they can tolerate water, but they don't require it for life. Um, so a lot of times these will be insects that live at the stream edge, or they'll live in the wet soil near the stream, which if the stream floods or the lake fills, maybe they get submerged for a couple weeks and then it recedes, but they can
Hmm.
tolerate the wet, but they don't need it, whereas an aquatic species needs that water to survive and a terrestrial species can't survive in the water. But there's terrestrial species that live in dead wood, dead decaying wood, so I'd imagine they have a certain set of characters that are unique. But it would be cool if you have one that's in decaying wood but still has a trait that's considered aquatic. Then what's happening there? Why does it, And so there's a lot of questions that fall out of that.
Cool.
And then I'm doing a couple other side projects too, so it's kind of all just going now. A smaller barcoding project with crane flies, another genetics project, and then some peripheral stuff that I'm not there yet, but I'm getting there. I used to work on cuckoo wasps, which I feel like I talked about the last time I was on, maybe.
Yeah. Well,
Um,
I think you've been on at least twice before.
that's
One where
true,
we
yeah.
just wandered over and was like, "Hey, Bob, what's happening?"
I remember that
That
now.
was
Yeah,
fun. And then there was
I
the one
forgot.
where we did the full broadcast of your presentation to the American Entomological
Oh yeah,
Society
that's right. That's right.
on
Yeah.
cuckoo wasps. I
Oh
think on
wow,
your
it feels like
master's work.
yeah,
That
yeah,
was a while back.
yeah. So yeah,
So welcome
so
back
I'm
again. Great to have you
working on
here.
those a little bit, and
Cool.
another project that I can't really talk about right now, but again, maybe when I come back, I'll talk about it. So
Oh yeah. Yeah. Cool. Um wait, so that's a lot of you're in your fifth year and um like how when do you think you'll be defending and everything out of
my
curiosity?
my goal is 2023
Okay.
next year. So it's going
Cool.
to be a big push, but we'll see. Like probably late in next year, I imagine I can be done. We
Mm.
the big barcode, the big molecular project we're doing, I was meant to go and collect all of the specimens we would need for it, but COVID shut all that down. So in 2020, when we were all stuck at home, I did a little mini Twitter campaign and asked people to,
Oh
I would send
yeah.
them vials of ethanol and they would collect for me and record where they collected them, the data, and put them in the vials and then send them back to me. And this was a raging success. I was so shocked. I expected one or two of my entomology friends to be like, "Yeah, I'll send you a couple." We got over 30 responses from uh, over a dozen states. We actually also got international interest, but at that time nobody was shipping anything. COVID again. So, but we got hundreds and hundreds of specimens from all over the U.S. I'm still actually processing them 2 years later. There's
That's
just
so
so many,
awesome.
and they're still coming in. Like, people still send me stuff. And now that things are open, we have two people who are helping us in Australia. So we're going to get some Australian topulids that we would
Cool.
definitely not have gotten anyway. So
And
I,
I'm
I
hold. Oh,
oh, go ahead. Sorry,
I was
no,
gonna
go
say,
ahead.
and I'm holding out the small hope that when I finally am able to go to Borneo for the research, that maybe you and John can come over and do
Oh,
some
that would be so
collecting in the
cool.
forest there too. I definitely saw a bunch of topulids at like the base of trees, sort of just like bouncing around, doing their
Yep.
thing.
Oh, that's so cool. I would love to go. I've
Yeah.
never been outside the U.S., so I would love
Really?
to. Yeah, never been outside the U.S. Well,
What, Canada?
no, I mean, I was in Hawaii, which is still part of the
Oh,
U.S., but that's like the
yeah.
furthest from home I've ever been, kind of.
Okay.
But
Then
yeah,
go
never
bigger, go
outside
big
the
then with
borders.
this,
Yeah.
hopefully. It's definitely a whole other world over there. Yeah, on the other side of the planet. Yeah, we'll be in touch about we'll see
Oh,
how
please let
things
me know.
go.
Yeah, that'd be cool.
Still in a holding pattern.
Steve? What was your question? I missed
It's
it
a
up
silly
here.
question.
Ah, let me see here if I can find it.
I can read it
Oh,
out
the
loud.
coolest person
Oh.
who's in it. And why is it Issa? Because Issa's the best.
Too. I don't know. Anyway,
Issa inspires me to be a better entomologist. That's what Issa does.
oh,
That's why she's the best person
thank
in
you
our
guys for
department.
being a part of our wonderful community too. It's all I get the inspiration from you guys too,
Ah,
and hearing your stories and your drives and your passion and commitment to exploring and expanding the boundaries of entomology, and also your willingness to join me here
well,
and
you
to
pushed
share
me
it.
to reach out and be a SciCommer too, so I'm trying.
Yeah.
Maybe
Um,
I'll have a stream here before too long.
yeah. Well, you're welcome to come back and share like updates on your work whenever you want to. And because it's really wonderful to have you on, and perhaps when I'm up in town next time, um, we can say hello in person and whatnot. So, oh, Terrence. I guess mini recap for the bug scope. One, I did like a couple-minute intro about how the word of the week was. You'll find out if you watch the beginning. Um, and then we had like a 20-minute technical difficulty bug. So unless you like that kind of bug, skip it. And then uh, Bob, who studies wing wing interference patterns on crane flies, which is related to our wordle of the week this week. That's why he came on. Talked all about his project, which has some really dazzling images and cool findings and whatnot from a feature that was noted in the past but not really explored until
Yeah.
Bob came along, and here we are. So,
And everybody, thank you for joining me today and tolerating my difficulties. I appreciate it, and thanks for joining me. I love sharing this work. I'm so excited that other people are excited about it. But if you have any questions, Issa, did you give them my Twitter handle? I'll put it in the chat. If
yeah, go ahead
anybody,
and put it in the chat.
yeah, if
Yeah.
you have any questions later on, that's my Twitter handle.
Or
Shoot
if
me
you
a
want to
line.
send a crane fly to Bob
Yeah,
as well.
yeah. If you want to catch crane flies, please
Yeah.
send me a message. I will absolutely
Yeah.
do that.
Be
Message Issa if you can't get to me. That's fine if you don't
yeah.
have Twitter.
Cool.
She'll give you my email.
All right.
But
Thanks,
anyway, thank you all
everybody.
so
This
much.
was
Yeah,
great.
thanks.
So much fun. Thank you, Bob.
Thank you, Issa. All
Yeah,
right, we'll
take
talk to you
care and
soon. I'll
have a
join
great
you
weekend.
hopefully before the end of the year. Maybe I'll have some new stuff to tell you.